Hi, myself I am Dr. Rangarao, working as Associate Professor in the Department of Biotechnology. So, today topic is conformation of glucose structure, linkages and glycosidic bond. So, this topic myself and my colleague Dr. Srikanth, we both are sharing. So, this topic covers conformation of pyranose and furanose ring, structure of glucose, linkages and glycosidic bond. Conformation of pyranose and furanose ring. So as we know that glucose is the most important molecule for living organisms, and it contains aldehyde group and hydroxyl group. The hydroxyl group can be distributed at the right hand side or left hand side. Glucose is called D-glucose or L-glucose according to the orientation of the carbon 5. Carbon 1 and carbon 5 react and then it will form the glucose ring. OH at the right side becomes lower, then we call alpha. On the left hand side becomes beta. This is example for glucose structure. Here you can see alpha D glucose. Here you can see D glucose. And the next one alpha D glucose. You can see here carbon 1 and carbon 5. Here carbon 1 react with the carbon 5 and it will form the glucose. And you can observe here OH is right hand side. So this is alpha position. So alpha D glucose. And then next one beta D glucose. Next one alpha D glucose and then beta D glucose. But this one is Fischer projection and Haber projection. Alpha D glucopyranose, beta D glucopyranose. Alpha D glucopyranose you can see on carbon number 1. Carbon number 1 you can see here OH is lower hand side. And beta D glucose you can see here OH is upper hand side. Upper hand. So that's what we call alpha D glucose and then beta D glucose. Glucose is a monosaccharide which is having C6H12O6 carbon, 6 carbons, 12 hydrogen and 6 oxygen molecules, whose 5 hydroxyl groups are arranged in a specific way along with its 6 carbon back. You can see here structure of glucose here. Carbon 1 aldehyde group on the carbon 1 aldehyde group. Carbon 2 you can see the right hand and left hand. Right hand hydroxyl group and the left hand hydrogen. And the carbon number 2, carbon number 3, carbon number 4 and 5 you can see how the arrangement hydrogen and then hydroxyl groups. In its floating open chain form, the glucose molecule has an open and unbranched backbone of 6 carbon atoms, carbon 1 through carbon 6. Each of the 4 carbons, carbon 2 through carbon 5 is a stereo center. We call carbon 2 to carbon 5, so is a stereo center. These 4 parts must be in a specific position, 3-dimensional arrangement. These are the 4 parts must be in a specific 3-dimensional arrangements. When the molecule is drawn in the Fischer projection, the hydroxyl groups on C2, C4 and C5 must be on the right hand side, whereas C3 must be on the left hand side. You can see here in the structure. Glucose generally represented three ways. One is a straight chain form, and the second one is alpha D glucose Hayward projection and alpha D glucose chain form. This is chain form structure. In aqueous solution, 99% of glucose molecules exist as a pyranose. The open chain form is limited to about 0.25% and furanose exists in negligible amounts. However, formula for sugars, pyran ring. Pyran ring, if you see the pyran ring structure here, it contains 1, 2, 3, 4 and 5 carbons with one oxygen group. And alpha D glucopyranose, that means here alpha D glucopyranol. This looks like a pyran ring. That means 6 carbon atoms with the 6 carbon atoms. 1, 2, 3, 4 and 5, 6 carbon atom and one oxygen. And you can see here the structure looks like a glucose and then this is a pyran ring. You can see the structure looks like a pyran ring. So that's what alpha D glucopyranose. You can observe here on carbon 1, OH is lower position, lower hand side. So that's what we mention alpha D glucopyranose. Here furan ring. This is a furan ring. The furan ring contains 1, 2, 3, 4 carbon with one oxygen. And fructose, alpha D fructofuranose. The structure looks like a furan ring. And then you can see on the carbon position 1, OH is up lower side. So here alpha D glucofuranose. The structure is a fructose structure, but the structure looks like a furan ring. So that's what alpha D fructofuranose. The reaction between C1 and C5 creates a molecule with a 6-membered ring called pyranose. The reaction between C1 and C4 creates a molecule with a 5-membered ring called furanose. In either case, each carbon in the ring has one hydrogen and one hydroxyl attached, except for the last carbon C4 or C5, where the hydroxyl is replaced by the remainder of open molecule. The ring closing reaction makes carbon 1, that is we call chiral carbon. Since it's 4 bonds, the carbon is having 4 bonds. So the carbon 1 is having 4 bonds. That is one is hydrogen and the other one is hydroxyl group, and next one carbon and the other one is ring oxygen. The C1 is completely made up completely. Actually, this is a bond. It contains 4 bonds. The 4th bond is associated with the C1. These 4 parts of the molecule may be arranged around C1, that is we call anomeric carbon, in 2 distinct ways, designated by the prefixes alpha, either alpha or beta. When a glucopyranose molecule is drawn in the Haworth projection, the designation alpha means that hydroxyl group attached to C1. The designation alpha means that hydroxyl group attached to C1, and the CH2OH group at C5 lies on the opposite sides of the rings plane, that we call trans arrangement, while B means that they are on the same side of the plane, so we call cis arrangements. Coming to linkages, when these linkages will form, when aldehyde can react with alcohol to form any acetal. Aldoexers such as the glucose. Glucose, the carbon 1 aldehyde in the open chain form of glucose react with the carbon 5 hydroxyl group to form intramolecular hemiacetal. The resulting cyclic hemiacetal, a six-membered ring, is called pyranose because of its similarity to pyrene structure. This is an aldehyde react with alcohol, then it will form hemiacetal. Coming to ketone. A ketone can react with alcohol to form ketone. Carbon 2 ketone group in the open chain form of a ketone hexose such as a fructose can form an intermolecular hemiketone by reacting with either the carbon 6 hydroxyl group to form a 6-membered cyclic hemiketone or the carbon 5 hydroxyl group to form a 5-membered cyclic hemiketone. The 5-membered ring is called furanose because its similarity furan. This is the structure here. This is the reaction. A ketone react with alcohol to form hemiketone. Glycosidic bond. The glycosidic bond, when actually it happens, so one molecule of sugar react with another molecule of sugar molecule, then it will form disaccharide with glycosidic bond. A glycosidic bond is formed between the hemiacetal or hemisaccharide and the hydroxyl group of some compound such as an alcohol. So glycosidic bond is formed between the hemiacetal or hemiketal group of saccharide and the hydroxyl group of some compounds of such as alcohol. A substance containing a glycosidic bond is a glycoside. Two monosaccharides can form a disaccharide with a glycosidic bond. This is the structure of glycosidic bond here. One of the sugar molecule react with another sugar molecule, then it will form glycosidic bond. This is here example alpha 1 comma 4 glycosidic bond. This is a small video that how this glycosidic bond will form. You can see here this one, the glycosidic bond here. This is one of the sugar molecule and this is another sugar molecule. It is react with it together and then this will form disaccharide. You can see 1, 2, 3, 4, 5, 6 sugar. This is a kind of, you can see the sugar molecule and this is another sugar molecule. You can see here and the glycosidic bond can be maybe sometime it alpha or beta with respect to the numeric carbon. Okay, you can see here alpha 1 comma 4 disaccharide and the other one is beta 1 comma 4 disaccharides. You can see there are two sugar molecules. Okay, it will form disaccharides with the glycosidic bond. How glycosidic molecules join together? So when the condensation process happens, then this glucose molecule, then it will form maltose. For example, maltose structure you can see here. Glucose actually react with another glucose, then it will form maltose. When it is formed the maltose, there you can see that between there is a linkage alpha 1 comma 4. Two sugar molecules are linked by condensation process. Then it will water molecule come out and then the two monosaccharides are linked by a glycosidic bond and the molecule share an oxygen atom. During this condensation process, the water molecule released and it will form. So thank you all. This is today's lecture and we can see in the next class about some disaccharides and then oligosaccharides and some of the how it is it will form the linkages and then how this glycosidic bond linked to the various structures. Thank you all.